All right, I think we're going to go ahead and get started. And several of you made the comment during the break that you're having trouble hearing. And I totally concur. I'm in the second row, and I guess it's my aging auditory system, or possibly the acoustics in the room, because we do have the volume all the way up on our mics. So we're just going to project a little more for you. So thank you for your patience, but I totally agree. We're going to try to do better. So I hope you all had a chance to vote on your favorite NeuroArt and visit the neuroactivity booths. It's now my pleasure to continue our program by introducing the next speaker, Dr. Carina Oehrn. Dr. Oehrn is an assistant professor of neurological surgery and a core faculty member at the Center for Neuroscience. She is our most recent hire. She just came on board in October. She studies non-motor symptoms of Parkinson's disease using intracranial neural recordings in humans and adaptive deep brain stimulation. Early in her career, Dr. Oehrn investigated brain networks underlying Parkinson's disease, and a key focus of her work is developing novel brain stimulation therapies for patients with neurological and psychiatric disorders. Her research explores how brain signals can be used to adjust deep brain stimulation dynamically at different times of day so that she can adjust the stimulation for individual patients' needs. This would affect not only the movement disorders in Parkinson's disease, but also non-motor symptoms like depression and memory impairments. By combining recordings from deep brain structures with innovative stimulation techniques, her work bridges the gap between cognitive neuroscience and clinical research, which is really exciting. So today she's going to share her research on novel deep brain stimulation therapies for Parkinson's disease, one of those new, novel, innovative approaches that helps us celebrate this in our 10th anniversary of neuroinnovation. Dr. Oehrn.
Thank you for the very kind introduction. Hello, everyone. Can you hear me well? If I just stand here? Okay, perfect. Perfect. So before we get started, happy International Women's Day, everyone. So let's give it up to all the amazing women in this room and around the planet. And yeah, thank you so much for having me. It's such a pleasure to be here and to speak to you about this very exciting topic of emerging deep brain stimulation therapies for Parkinson's disease. 2024 has been a very big year for deep brain stimulation research because we had a couple of major breakthroughs in the field, and I'm very happy to share a couple of them with you today. So Parkinson's disease is the second most common neurodegenerative disorder and affects more than 1% of the population above the age of 60 years old around the planet. So it is not uncommon. And most of you probably know that Parkinson's disease is associated with an impaired movement characterized by slowness, stiffness, and around 50% of the patients also have tremors. And this is an image that you frequently find in the literature and in the media of a typical patient living with Parkinson's disease. So you can see the forward tilt of the trunk and the rigidity and trembling of the hands and head and the shuffling gait. But it's also very, very important to emphasize that Parkinson's disease can come in all shapes and forms and at every age, actually, and can range from a slight tremor of the hand in combination with a cramping of the foot to severe, consistent gait impairment. And this neurological disorder is associated with a dysfunction of so-called basal ganglia cortical circuits. So basal ganglia are structures deep inside the brain. And we have found that neural signals at very distinct frequencies, which we call the beta and the gamma frequency, are associated with particular symptoms in Parkinson's disease. So now you may ask, how do we know that these signals play a role for the symptoms? How can we record neural activity, brain activity from deep inside the brain of patients? And we can do that when patients are being implanted with electrodes for deep brain stimulation. Deep brain stimulation is one therapeutic option for Parkinson's disease. We'll talk about the therapeutic aspect in a second, but let's just talk about where these signals come from for a moment. Oh, this is going back, sorry about that. So we can either record the brain activity in the intraoperative setting while patients are being implanted with the electrode. So here you see an example of a patient speaking to me while we're performing the surgery, because they're usually awake while we do this. And then some of the patients are willing to do tasks with us, so you can learn more about what the brain is doing. The second option is sort of a perioperative time, so a few days after surgery, while the deep brain stimulation device is not connected yet to the electrodes. And during that time, we can record activity and we can learn about the brain. But a great new development in DBS clinical treatment, but also research, is that we now have devices that can record this brain activity chronically. That means even after the patients have left the hospital, months and years later. So that gives us a great opportunity to study these signals in real life. What kind of properties, what kind of characteristics do we look for in these signals? So a little bit more than 20 years ago, the first papers, the first publications emerged showing that inside these neurosignals, we can find rhythmic patterns at very distinct frequencies, as I mentioned before, that predict some symptoms of Parkinson's disease. So you find these rhythmic oscillations of neuronal activity, and that reflects the synchronized activity of many tens and hundreds of thousands of neurons in the brain. So at the moment, unfortunately, we can't cure Parkinson's disease yet, but we can really help to alleviate the symptoms very well for quite a long time. And so in the beginning of the disease, medication is very effective for Parkinson's disease patients, but after a while, it gets a little bit challenging to consistently control the symptoms throughout the day. And this is when deep brain stimulation becomes a very effective treatment option. And during deep brain stimulation, or during this treatment, an electrode is implanted deep inside the brain into, like most commonly, a brain area that is called the subthalamic nucleus, the SDN, and is then connected to a brain stimulator that is implanted in the chest, so you can't see it from the outside, and the cable is implanted underneath the skin. And then this device generates activity continuously and applies this in this brain target. And after the electrode has been implanted, the neurologist then still can optimize the stimulation in the patient's brain by, for example, changing how much stimulation you apply, how big is the amplitude, and also where exactly in that target brain region do you stimulate, depending on the patient's symptoms and also side effects of the stimulation. And just to give you a little example of how effective stimulation can be, just want to show you the effect on tremor, for example, on the left without DBS and on the right with DBS on. And this is a patient with severe gait impairment who received deep brain stimulation. And you can really see that this can have an immense impact on patient symptoms. But as we mentioned earlier, when I talked about medication, the symptoms of Parkinson's disease patients can fluctuate throughout the day. They usually fluctuate throughout the day. And they can experience phases when they feel really slow and the symptoms break through. And then there are other times when they get side effects, for example, of their medication, and they become hyperactive. So deep brain stimulation can help to reduce these so-called motor fluctuations. But how great would it be if we had an option to reduce stimulation when they're hyperactive, when they need a further reduction, and give them a little boost when they need it, when the symptoms break through throughout the day. And that is the whole concept behind adaptive deep brain stimulation. So the idea is that we continuously track a neural signal, a brain signal, that reflects the patient's symptoms, and then we adjust the stimulation in response to this neural activity. So according to the patient's needs at a given moment. So just to illustrate that in a little video, you see the patient here. Parkinson's disease causes slowness and rigidity in this patient. And then the electrodes that are implanted then sends an abnormal brain signal related to the worsening of symptoms. And then the device does its thing, a certain threshold is being crossed, the stimulation is then adjusted in response to the signal, and then in this example, increased in that target brain region. And then the patient feels a lot better, and the slowness is gone. So there are several studies now that show that this is feasible in the laboratory, and that it can either reduce battery consumption or even improve symptoms better than the conventional deep brain stimulation. But all these studies have been done in this perioperative settings, right? So like just a few days after the surgery has been performed. And so this is why they have several disadvantages. So they're limited to this laboratory setting. They're, as you can see in the table, relatively short recordings, right? They're limited to the off-medication state. So it's still unknown if these effects are true or hold true when the patients are cycling through their medication states. And these trials were also just focused on one type of brain signal. This is a very specific research comment, but the neural signal used was identified while stimulation is off. But in real life, we want the patients on stimulation the whole time. And we know that turning on the stimulation actually changes the neural signals. So it's important to study them like in the conditions we want to use chronically. And one of the biggest drawbacks is actually that in these trials, as this was just a few days after surgery, the control condition they compared it to might not have been quite fair because it actually takes months and sometimes years to optimize the brain stimulation, the conventional deep brain stimulation for patients. And so just a few days after surgery, where the patients also still have, side effects from the surgeries, such as the swelling of the brain, might not have been representative of what we will find a few months later or a year later. So you may now ask, so okay, the first paper on this was published in 2013, right? So why don't we see this in the clinic yet? Why is this not a treatment for Parkinson's disease patients that everyone is getting? And one of the reasons is that we just didn't have devices that were capable of doing this, of sensing brain activity while the patient is being stimulated in real life. And so around 7-8 years ago, the first devices came to market. They were first available to researchers, and now they're actually available to clinicians as well, that were able to stream brain activity to a tablet via Bluetooth that the patient carried with them. So we had access to these devices, and the patients who have been implanted with them chronically, and they could then stream their brain activity while they were just going about their normal daily lives. So we could record from their brain in the laboratory, in the clinic while we saw them, but then also while they were working, hiking, traveling. One of them was a researcher themselves, and they went to conferences and streamed their data to their tablet that they just had close to them. And this also enables us not only to identify the brain signal that is actually responsible for the symptoms in real life, but also develop adaptive algorithms for real life and then test them. So we wanted to bring adaptive deep brain stimulation to the real world, so sort of from this perioperative setting to this long-term testing. And for us, so in the beginning, or for this trial, let's say, because it was a very, very intensive trial for patients, we enrolled 4 of them. They worked at least as hard as we did. And they had electrodes implanted in this deep brain region that we spoke about, the SDN, and then also the cortex. So like a more superficial brain region. And then we made life a little bit more difficult for ourselves compared to other studies, because for us it was really, really important to actually tackle the symptom that was important to the patients, and not some artificial definition on some rating scale. So we asked every single patient, what is the symptom that actually bothers you? What is the symptom that breaks through throughout the day? What should we tackle? And actually 3 of our 4 patients said it was the so-called bradykinesia, so the slowness. So they had phases of slowness breaking throughout the day. And there was one patient, the one who travels a lot, who had a cramping of the foot that really made it difficult to walk during certain times. And then we assessed their symptoms with digital motor diaries on their phones. And we also had smartwatches that could track certain motor symptoms. And so the timeline of our trial was that we first gave an independent neurologist as much time as they wanted, to optimize the conventional deep brain stimulation. So this took from between 11 months to 31 months. So they really had all the time they wanted. And as soon as the stimulation was optimized, we then started with our adaptive trial. And in the second phase, we then recorded their brain activity in the clinic and then also at home, and try to find out which brain signal is associated with the symptom that bothered them. And in the third phase, we then developed the adaptive algorithms. And in the fourth step, we then tested the adaptive deep brain stimulation to the conventional one that had been optimized before in a randomized and blinded way, where they were just going about their normal daily lives. And every few days, between 2 and 7, depending on their schedule, they could click a button on a tablet that just said, switch my stimulation, and they didn't know what it switched to. And this is just to illustrate this brain activity that we were streaming. So what does it actually look like? And I really wanted to show that to you because this is very cool for us neuroscientists, because we hardly ever get to observe brain activity from humans just going about their normal daily lives. And this patient streamed brain activity from 7 a.m. to 7 p.m. And you might be able to see that there is this particular brain signal, sorry for the guys on the other side, at around 65 hertz. So the x-axis is the time of day, right? The y-axis is the frequency of the signal that we observe. And we saw in every single patient that there was this one signal that always increased when they took their medication. So the red line indicates when they took their medication. And then whenever their symptoms were breaking through, so these are the black bars, right? And the symptoms are related to when they feel bad, when they feel off. The signal wasn't there. And of course, we did statistics and everything. This is just an illustration. So really consistently found that this seems to be a signal that is good for their movement. Whenever the signal was there, they felt good. So this is why we use this signal for the adaptive deep brain stimulation. And on top, you can see the neural signal fluctuating throughout the day, and in red, the medication intake. And in the bottom plot, you see the stimulation and how it adjusted. And because this signal is good for movement, we dropped the stimulation when the signal was high, let's say in this example to 2 milliamps, and we increased the stimulation, so gave the patients a little boost when the signal was low, right, when the symptoms were coming back. And just as an example, let's say in this patient, the conventional stimulation would have been 3 milliamps, right? With the adaptive, we can then cycle between 2 and 4 whenever the patient needs it. And now to the most important slide. What did we find? What were the results? So we found that the adaptive deep brain stimulation cuts the time with symptoms in half in all of our patients, and it improves quality of life. Apart from one patient who always had a high quality of life, didn't matter how bad the symptoms were. But in blue, you can see the conventional stimulation and how much time. So on the y-axis, you see the duration of symptoms throughout the day. And in red, you see the adaptive stimulation. So these are very, very promising results. And now many centers around the world are starting to replicate them and enroll more patients into the trials. And if you're interested in reading a little bit more about this, one of our 4 patients was kind enough to share their story about their Parkinson's disease symptoms and participating in our trial. And you can read about it in the New York Times, published in September. All right. Now, switching gears to another big need for treatment options in Parkinson's disease. So we talked a lot about the motor symptoms in Parkinson's disease, right? But the neural networks involved in Parkinson's disease that I show here do not only process motor, but also cognitive and emotional information. And this is why it's not surprising that patients also develop cognitive and affective symptoms, such as deficits in working memory or inhibitory control, anxiety, apathy, depression. And they are quite common, as you can see, right? And there are several papers now showing that these symptoms actually affect quality of life more than the motor symptoms. So this is really an area that has been a little bit neglected in research so far because everyone always focuses on movement, and we hardly have any treatment options available. So it's very important to study this field. And so there are a couple of publications now showing that during cognitive processing in Parkinson's disease, you see a signal at a slightly different frequency. So at around, like, let's say between 6 and 8 hertz in most patients. So this always increases when they do certain cognitive tasks. And interestingly, one pilot study actually tried to stimulate them at this lower frequency compared to the frequency that we usually use to improve their movement and see if that changed their behavior, and patients actually performed better. So this is very promising. So this is 5 hertz stimulation in that low frequency compared to 130, which we usually use in the clinic to help them with their movement symptoms. And so we started a study asking whether we can generalize these effects to other core cognitive functions. And one of the biggest problems Parkinson's disease patients have that you can find across the literature is an impairment of working memory. And so we performed, we recruited 20 Parkinson's disease patients with electrodes in the SDN. And then we asked them to perform a working memory task that pretty much consisted of seeing digits and then having to reproduce the digits, so like a phone number. And we also did short motor tasks with them. And we did this under 5 stimulation conditions. So off stimulation, then the general, the high frequency stimulation we usually use in the clinic to improve their movement, and then also beta frequency stimulation. And we found a very interesting double dissociation between effects on cognition and movement. So when we look at cognition, so in this case, the y-axis is working memory performance, or the change of working memory performance, and more is better. So we found that exclusively theta frequency stimulation, so at that frequency where we observed the signal, improved working memory performance, whereas the other frequencies didn't affect it. And when we looked at movement, and in this case, a lower score is better because it measures how impaired the movement is, we found that this high frequency stimulation in the gamma frequency range actually improved movement, which is not surprising because this has been used for many years in the clinic, but the lower frequencies didn't impair movement or didn't affect movement. And this is very promising because the low frequencies didn't affect movement negatively, but improved cognition. So maybe we can use them together in combination, so the lower frequency to improve cognition and the higher frequency to improve movement in the future. And now there are several clinical trials going on. One of them is here at UC Davis to test whether this could work and chronically applied improve the symptoms of patients. So in summary, I showed you that adaptive deep brain stimulation is a promising new treatment for Parkinson's disease, and adaptive DBS that responds to specific brain activity patterns improves motor symptoms and quality of life in a real-world setting. And low-frequency stimulation in this deep brain region in the SDN enhances cognitive functions compared to standard clinical stimulation. So overall, I hope I've shown you that understanding brain activity linked to these symptoms really helps us to fine-tune the conventional DBS and also develop new adaptive DBS approaches. And with this, I would like to thank my lab, who has worked mainly on the second study about working memory, and then also my amazing collaborators at UCSF. And if you have any further questions, please feel free to ask after the session as well, or write me an email anytime. Thank you very much.
All right, thanks for a great talk. My grandfather had a deep brain stimulator for his Parkinson's, and as my mother described it, he was gone for 10 years, 15 years, and then he came back one day. So it's really interesting to hear about. I'm curious about, it appeared in the diagram, there were 2 places for monitoring, and then there were the deep brain stimulators. Is there any monitoring at the site of stimulation, or is it just there? And then how's that being processed? Is that going through is that being processed and filtered to check for specific frequencies, or is it more of a machine learning process, and then, well, I'll save that. Yeah.
Amazing questions. Thank you so much. Yes, I somehow brushed over this a little bit, right, where we implanted the electrodes because I thought maybe we don't have enough time to go into detail. But exactly. So we stimulated in this one deep brain region, the SDN, and we recorded from this deep brain region. So yes, we recorded from that implanted deep brain, from this deep region and the cortex. The cortex is usually not implanted. This was really specific to the UCSF trial where they do this chronically. But in our trial, we actually found that in this more superficial region, the cortex, we found better signals that were actually a stronger predictor of the symptoms than in this deep brain region. We don't know yet whether this is because, I mean, these are some of the first devices on the market, right? Maybe they reject artifacts, the artifacts of stimulation quite well, but maybe it's related to some of these artifact rejection methods. But yes, we recorded from these two regions, and now, but we also found like this high frequency signal in both, right? And so now many centers around the world who don't record from the cortex, just from the target brain region, are replicating this, that this is still like a promising neurosignal that we can use. And what was the second part of the question again? Sorry.
Yeah, I was just wondering. I'm curious about like oscillation in neural networks,
Yes.
and I'm just wondering. It would seem that monitoring at the site of stimulation would be important to stop like phase cancellation, but I don't know if that's an important consideration.
Yeah. So everything that we record is being processed on board of the device. Well, a lot of the analysis that we've done, we did offline, downloading the data, processing it, and then seeing what is the best signal. But so when the device is performing adaptive deep brain stimulation, it's sort of doing the calculations based on a neural signal online, like just on the fully embedded on the device. And the device is able to process power and not really phase information or any more complex measures. So this is why we focused on power only. So this is a measure of how much of this rhythmic pattern is in the signal. And we took a completely data-driven approach, kept an open mind. We didn't restrict it to any frequencies. And actually, what we found is what the frequency a lot of papers have published in the laboratory wasn't related to the symptoms in real life at all. So a lot of papers have shown that beta oscillations, like at a medium frequency, are associated with the motor symptoms. But when we tested it in real life, beta oscillations, they went up and down unrelated to their motor fluctuation. So I think this really also underscores that it's really important to record brain activity in real life and associate it with real life functioning.
I have a close friend who's had Parkinson's for like 10 years now, and he had stimulation put in about 4 years ago. And I never thought about this, but in the depiction of the guy cutting a carrot, okay, so does the activity send a message to the brain of what the activity is and then sends a signal back to do it better? Is that what happens?
That's what happens in what we did. So this is not like a standard treatment yet, the adaptive deep brain stimulation, right? So at the moment, most patients are implanted with devices that go one way, stimulation only. It's not sensing what the patient is doing. It's not sensing the needs of the patients. So this is really new about what we developed, of course, working with a company. But
Because he goes back about every 3 months to have things reprogrammed or adjusted.
Yes.
And I've taken there, but I've never been in the meeting. So in doing that, would the doctor say, what activity do you see slowing down, or and then makes adjustments for that?
Exactly. Yeah, your question is so important, and thank you for bringing this up, because I didn't underscore this enough, I think, exactly. But the standard deep brain stimulation at the moment, patients have to go back to see the doctor every 3 months, let's say, right? And the doctor then spends 1 hour with them fine-tuning and asking them what the symptom is. So this is really the big advantage of the adaptive deep brain stimulation. It does it automatically. It responds to your brain activity, to your needs, and then goes up and down in stimulation, depending on how you feel throughout the day, right? You might not always need the same level of stimulation. With the standard deep brain stimulation, it's always the same level.
because his cognitive and emotional seems to be very good. But I think part of that is that he socializes a lot with those activities, you know, having discussions, playing games.
That's important.
You know, and so my question about what stimulates what, you know, that he's receiving those signals and then the brain is somehow either making them work or not making them work.
If you like, we can talk a little bit further, maybe after this session, about this. Thank you.
Hi, I have the microphone. I have, here.
Hey, sorry.
Thank you for this. I have a bunch of questions, but here are three.
Yes.
These probes in your brain, how long do they last before the brain says, this is something shocking me, I need to get rid of this? The second question is, you've got frequency and you have voltage, right? So I'm curious how you figure out the proper voltage. And the third question is, the frequencies, has there been studies done on applying the frequencies through different modalities like light, sound, or,
Yes.
physical?
Thank you very much. Three very good questions. The three very important questions. So to your second one, how do you find out which frequency and amplitude? It's really trial and error at the moment. So the patient comes, sees the clinician, and then we spend a very long time with the patient and try all kinds of different combinations. But this is also mainly why clinicians have just settled on a frequency, that across patients, this is the one that mostly seems to work, and if it doesn't work, then you deviate from the frequency and maybe try something different, right? But it's always around the range of 130 hertz. And then the amplitude and where exactly we stimulate is then really based on sitting with the patient, increasing the amplitude step by step, seeing if the symptoms improve, seeing if side effects occur, and then doing this at every single one of these electrodes that you have implanted. So it's a lot of work. It's a lot of work. That's why we're all working on automating this in a way, right? Finding brain signals that tell us where the best effect will be and things like that. So that's how we usually do it. And with the different modalities, yes, that's incredibly interesting because a lot of them are non-invasive, right? So that's very promising. So we did one trial, we performed one trial, and I think we're also going to extend it a little bit with patients where they listened to sounds that were that contained this frequency that was beneficial for movement, and we found some good effects on tremor only, resting tremor. But we didn't really have a large population to understand why and why exactly that symptom and what is going on in the brain. So we will study this further. And exactly. So the first question was again, sorry, I think it's been a long day. Oh, yes. So the probes are being implanted forever. The patient keeps them for the rest of their lives. And on most symptoms, they have a really good, stable effect over time. But of course, there's also still ongoing neurodegeneration, right? So the symptoms will get worse over time. And then the clinician has to adjust the stimulation in response to that and then increase amplitude every 3 months or so, right? There is one symptom that is a good example of what you're asking, because tremor, after a while, sometimes stops responding to deep brain stimulation because the system adjusts somehow, right? The other symptoms, they remain stable, but for tremor, then sometimes a stimulation withdrawal is done in the clinic, where they just turn off the stimulation for 3 days, and then it's effective again for, several months. So, but we're still, doing a lot of research about understanding why these are differently affected.
Last question.
Am I on?
Yes, you are.
I have 3 questions. The third one you're going to like.
I will be.
You guys are catching on.
Better as opposed to, you're right. You might want to turn my mic off. We came in a little bit late. I missed the preliminary. What studies preceded yours? Oh, this way? Oh, okay. What study or studies preceded yours, and number 2, how many trials have you conducted to reach the point that you're at now? That's 2. And number 3 is, you know what I'm going to be saying, how much of this work do you anticipate is going to be cut by you-know-who?
What was the third question? I just had trouble hearing it a little bit.
How much of the study will be cut by you-know-who?
How much of the study will be cut? Cut. Okay. Well, you know who is collaborating with a who's very interested in invasive deep brain stimulation. So who knows? But at the moment, we're still okay for funding for a little while, right? But of course, like, in a little while, it might run out and be affected by these cuts. So, but let's see what kind of research is going to be funded. But yeah, I think we sort of have to extend, our sources of support for this kind of research. And then the second one was, how many trials have we performed to arrive at where we are with the adaptive deep brain stimulation, you mean? This is so we only worked on this one, but there have been other trials, and they try to focus on this one brain signal and only on that brain signal that has been published in the laboratory. And they have like mixed effects. I think it's because we sort of used this data-driven approach and kept an open mind and said, like, let's just see what brain signal, is associated with the symptoms. We were successful in this case. So for the adaptive, it was really just this one. There is one going on by a company as well, which is helpful because they will bring this to the patients on a larger scale. And I think they will publish their results in a few months.
Thank you so much.
Of course. There was a first one where we can talk after the session if you like. Thank you.
Thank you.
Thank you.